Thank you, Marianne, for the introduction. I will spend the next 15 minutes to explore how we can use long-read sequencing to diagnose rare disease patients in routine diagnostics and to improve the diagnosis and to accelerate the diagnostics. If you're working in rare disease diagnostics, you know you need an entire zoo of methods to completely diagnose your patients, and even with that entire zoo, you're not achieving a diagnostic rate higher than 50%. But this means that maintaining all these different workflows, it imposes a logistic challenge. You have to maintain those methods, and the different samples have to undergo these different workflows. You may also have batching issues because certain methods like MLPA require a certain number of samples so that it's economic to perform the analysis. This typically leads to a stepwise approach. So you will perform one method after the other, which leads to a longer diagnostic journey of the patient. And depending how your healthcare system works, if you're performing the tests in-house or if you're sending them to another laboratory, this may impose additional costs for you. I listed also short-read sequencing assays at the bottom, and we were pretty successful with our software to replace certain assays in the laboratory by utilizing all the information that is contained in short-read sequencing data. We were very successful, for example, replacing MLPA with a CNV analysis from short-read sequencing. But it's clear that short-read sequencing has shortcomings, and we cannot replace all the other methods in the laboratory that you need for the comprehensive diagnostics. So the question is, and has been posed for quite some time now, long-read sequencing has so many unique properties and promises to perform comprehensive diagnostics. This should be the one test that fits all, and probably the group from the University Medical Center in Nijmegen in the Netherlands, from Alexander Heuschen, probably most advanced in that aspect. They recently published a study comparing standard of care with this zoo of methods to long-read sequencing using 1,000 patients, which is a comprehensive set of data, and they came to the conclusion they can certainly replace everything that they're performing so far in the laboratory with long-read sequencing. So the question is, why is not everyone using long-read sequencing yet? Right? Why has this not happened? And there are certainly a number of challenges. For example, the team in Nijmegen in the Netherlands, they have a large bioinformatics team. They develop their own software. But I think Oriane demonstrated that with the varvis software, you already have everything ready. It's one approved medical device. So far, you needed to invest into your own IT infrastructure to be able to analyze the long-read data. This is also something that the varvis software provides to you inherently. There's a question of cost efficiency, of course. I can tell that using our software for long-read sequencing is not more expensive than using it for short-read sequencing, so no cost difference there. And of course, we are very much anticipating the cost for long-read sequencing to come down as well. And I think even at this conference, there have been some announcements to that effect. As a clinical diagnostics laboratory, you always have to think about how are you demonstrating that the new method that you're now implementing in your laboratory performs as well as what you have done before. And since we are a medical device manufacturer and our device has been approved by a notified body, we can provide you with performance tests so that you can demonstrate also for your own laboratory operations that you're compliant with IVDR. So remains one last challenge that is frequently brought to our attention is kind of the interpretation burden. I received feedback from people in the laboratory now that they can see so much more using long-read sequencing. For example, thousands of structural variants that were not visible before. How will they deal with this in routine diagnostics? Will this not take much, much longer than the regular process with the standard of care? So this is what I'm going to focus on in the next minutes, and I will do this using two examples. I pick out fragment length analysis and array diagnostics to highlight how our software solves this the challenge of interpretation burden. Let's start with fragment analysis. I'm simply highlighting kind of the limitations of fragment analysis. Everyone who has performed this in the lab knows that there are certain size limitations. Very long, very much expanded repeat expansions cannot be accurately detected with this method. The sequence of the flanking regions are important. If you have SNPs in those regions, your primers may not bind and your assay may fail. GC-rich repeats are always difficult, a general challenge. It's basically impossible to resolve the exact composition of the repeats. Do you have modifications? Do you have interruptions of the repeat? And just by performing more and more long-read sequencing, we learn that those interruptions may be clinically significant. And you have a low sensitivity for mosaicism. And just as a reminder, this is what a fragment analysis looks like. This is a FMR1 gene. You have a decreasing range of peaks, and you basically have to count them manually. And you can see that, beyond 200 repeats, beyond the 200th peak, it's basically impossible to determine are there more peaks. You can only say, well, it's probably larger than 200 repeats. But as you can see, or as you know, this is actually the range where the pathogenic range starts for this gene. So it would be nice to have more information how much expanded it actually is, and have more confidence that it is actually larger than 200 repeats. So our software solves this using long-read sequencing data. First of all, we give you a quick overview of the composition of the reads for each allele. So you can see here the blue bars are the expected repeat structure. The gray interruptions are visible. We also indicate very clearly, there's a negative sample here, if the repeat expansion is in the benign range, in the intermediate range, or in the pathogenic range. And we also provide you with information about the age of onset. First, for all the reported individuals, and also what the typical age of onset for the disease would be. We provide context information from the bioinformatics analysis, of course, but we also provide links to disease websites if they're available, to relevant literature, gene reviews, etc. And we also provide specific information for your repeat locus regarding penetrance, more information about age of onset, the molecular mechanism, and also prevalence in the population. So the goal of all that is that you don't have to switch to any other source of information to properly classify the repeat expansion and to therefore accelerate the process. Now, and what I mentioned before, since mosaicism becomes probably more and more relevant, we also provide you a graphical display of a waterfall plot that shows you kind of the distribution of the alleles and how the length of these alleles differ. This is basically probably a very nice heterogeneous locus, but we can see from other patient data that repeat expansions may be very, the spectrum may be very broad, and the difference in length may be very broad, and we are exploring what is the clinical significance of this mosaicism. Now, switching to array diagnostics. If you think of the gold standard in array diagnostics, a high-density array of 1.4 million probes. This is a very powerful tool to discover chromosomal aberrations. But even this powerful tool has its limitations. For example, those probes, even if they are 1.4 million, they have a certain distance. So you can only detect variations down to a certain size. Typically, or most of the time, probes are located outside of coding sequence. So if a variation is adjacent to an exon, you still have to use other methods to confirm is an exon actually affected or not. You cannot detect inversions or balanced translocations. So again, even though it's a nice tool and powerful tool, you cannot cover everything in rare disease diagnostics. We try to replace array diagnostics with short-read sequencing. And now that we have a new updated benchmark from the Genome in a Bottle project, HGO2, the T2T assembly of that individual. We have really a very accurate source of information. What are the structural variants in that individual? And we can very nicely benchmark a multitude of methods with this assembly. And it's very clear, the results are very clear, even if you compare to the gold standard now in short read sequencing, the structural variant analysis from Dragon. Any long read sequencing technology will outpace your short read diagnostics. So long read sequencing is certainly, or will soon be, the standard of care for structural variant detection. We are complementing the structural variant detection with a CNV analysis that is based on coverage. So while the CNV analysis has certain shortcomings, it cannot detect certain structural variants, it complements the structural variant analysis, providing information about large aberrations, large copy number changes that the structural variant analysis is a little bit, or has not highest sensitivity for. And what does this look like in our software? I'm giving you kind of a curious example. This is an example in an Oxford Nanopore hereditary cancer panel. This panel is generated by using adaptive sampling, if you're familiar with the technology. So you have shallow whole genome sequencing across the entire genome, and in certain target genes you have higher depth long read sequencing data. And this is the result from our CNV analysis. The yellow dots are basically the probes of the CNV analysis, and across the entire chromosome it was wild type, 2 copy numbers. And in the BRCA2 gene here, we saw a very curious duplication, or basically 2 duplications, and a stretch of wild type of 2 copy numbers in between. And if you look at this from the structural variant analysis point of view, looking at the long reads, the structural variant caller actually detected a deletion in that area. So while the copy number analysis says it's wild type in that stretch, structural variant analysis shows you a deletion. And kind of the resolution to that curious problem is this was a reference sample from Seracare, where the manufacturer spiked in an amplicon containing a certain deletion. And since the amplicon at the rims of that stretch was a bacterial sequence, the structural variant caller did not detect anything at the edges, but only detected the deletion inside the amplicon in the middle. So both technologies are complementary, and we are very curious to see more patient data, how our biology is so variable. We will really like to see more examples where this is helpful. We try to give you as quickly an overview of the structure of those variants as possible. We provide simplified visualizations. And again, a wealth of annotations, for example, from variant databases, DGV and Decipher. But most importantly, we provide you with a ranking of those genes that are affected by a structural variant compared to the phenotype of the patient. So you can quickly rank the genes that you find from the structural variant analysis, and quickly see which might be genes that are linked to the disease of your patient. So in essence, so I think I made clear that at least for repeat expansion and structural variants, we streamlined the process, and we enabling now everyone, even those without a big bioinformatics team, to perform the analysis, to process the data very conveniently. And it's just now adaptation of the long read sequencing technology of the instruments, and maybe also of sequencing costs to come down, to establish this as the new standard of care for rare diseases. Thank you.